Our appearance can change, so can our behavior. We can get thinner, bigger, taller, healthier, less healthy, change our diets, change our clothes, but what happens beyond appearance? Genetic determinists would argue that unlike our phenotype, the genetic information constituting our genotypes are innate and fixed. According to this theory, genetic traits cannot be influenced by the environments. Thus, if there was a gene for a crime, for example, a criminal would biologically be determined to be a criminal. At this point, we have to raise the question, doesn't this assumption sound a bit too simplistic? Conversely to what genetic determinists argue, genetic information can be influenced by the environment. An example for this is the mental illness schizophrenia. The symptoms of schizophrenia have an impact on all areas of functioning and usually appear in adolescence or early adulthood. They include disturbed cognition, disorganized thinking, emotional repressivity, loss of concentration, drive, affection, delusional thoughts and interpretations, and auditory hallucinations. As all schizophrenia patients have different life stories, they can be differently affected by the symptoms of schizophrenia. The people affected by schizophrenia might have to face disturbing traumas, abuse, isolation, infections, or family history of mental illness, parental situations, arguments, divorce, or change in social class, or change of residence, particularly to a new city. Furthermore, interestingly, studies showed increased risk of getting schizophrenia with half-siblings and increased risks with younger rather than elder siblings.
Let's now examine the genetic basis of schizophrenia. Statistics show that the closer you're related to a schizophrenic, the higher your chances of being a schizophrenic. And adoption studies indicate that the link is not just environmental, but genetic. So let's look within the genome. We're now able to sequence and compare the genomes of schizophrenics and non-schizophrenics by a process known as genome-wide association study, or GWAS. Many GWASs about schizophrenia investigate common genetic variations. Copy number variations, or CNVs, are a form of sequence duplication or deletion. Studies show that deletions are more significant than duplications, and that larger deletions, such as an NDE1 involved in brain development, are associated with schizophrenia. In addition, single nucleotide polymorphisms, or SNPs, are when a sequence contains an extra or missing base pair, kind of like a genome typo. Studies show that SNPs associated with schizophrenia vary greatly between testing samples. In one study, the schizophrenics from Munich showed associations that those from Aberdeen did not. Also, many of the associations fail some statistical significance tests. What's more, the lowest p-value was for this SNP, which curiously is neither located in a gene nor strongly associated with an SNP in or close to a gene. But the homologue of the most associated SNP mediates signaling pathway involved in the nervous system. Although we can't blame schizophrenia on any particular alleles or coding sequences, we know that there are a few CNVs and SNPs that warrant further investigation.
Heavy consumers of cannabis at age 18 were over 600% more likely to be diagnosed with schizophrenia over the next 15 years than those who did not take it. Most research studies indicate that the risk is higher when the drugs are used by people under the age of 21, a time when the human brain is developing rapidly and is particularly vulnerable. People with a biological predisposition towards schizophrenia are at higher risk. Unfortunately, it's impossible to accurately identify this predisposition beforehand.
Schizophrenia usually shows its symptoms in early adulthood. For instance, men are diagnosed with the condition mostly between the ages of 16 and 25, whereas women seem to develop it in their late 20s. It has been suggested that the reason it affects young adults is due to a protein coded for by the gene DISC1, which helps guide new neurons to the correct site so that they can connect to other cells. When researchers shut off parts of DISC1 in mice, they found that the amount of dendritic spines decreased. Now, these connections are constantly broken and remade throughout our lives, but they happen most during our adolescence. So if a person has an aberration in their DISC1, then they're more likely to start showing symptoms in this age range.